Now, in the first section, I tried to place before you an overview of measurement system analysis. The focus there is on the error uncertainty associated with any measurement system. No matter how good your measurement system, there is always an associated uncertainty with it. Now, we also looked at that so-called variability has two components: the random component or the unknown component or the uncertain component, and the systemic component, and that is the error that can be corrected. We also saw where all the errors could possibly come from. We talked at length about specifications and raw materials and machines and environment and sampling, sample handling, sample storage, sample preparation, sample injection. But there's also one more element, and that is the analyst. Now, in this section, I'd like to highlight the role played by the analyst in the measurement system. I'm not talking about the analyst repeatability. That has become some kind of a routine exercise. You inject that many times, you calculate the mean, calculate the standard deviation, and if it is all right, it's all right. Be treated very lightly. But is it to be treated so lightly? Please remember that any instrument, no matter how sophisticated, is only an aid to measurement. The key element in measurement and interpretation continues to be the human being. But he's a human being. And because he's a human being, he's not a machine. You can't set the parameters and keep it in a state of control and you expect that the operator, analyst will continue to perform consistently. No, that doesn't happen. The analyst may be healthy one day, may be sick another day. May be happy one day, may be sad another day. May be full of energy in the morning, may be very weary and fatigued in the evening. He may be focused on his work one day and find that he is unable to apply his mind on another day. His mind is at peace on one day and is totally troubled on the other day. Now, he continues to make measurements, and those measurements have to be valid for anybody to take any kind of informed decision based on the results, numbers that are generated by the analyst. The difference between an ordinary person and a professional is the ability on the part of the professional, no matter which profession he belongs to leave his baggage outside the door. Once he enters his workplace, he has switched off from everything else and is focused. The level of focus, the level of commitment, and most important, the pride in performance, the determination to make sure that whatever work has come to that professional will be discharged or executed in the best possible way. That is what separates a professional from anybody else. And that is what is expected from you. Now, how do you know you are a good analyst? How do you show that you are the best analyst? There is only one way, and that is generate data. Generate data to show your consistency in the work that you do. And show that the variability in your work, the agreement, the mutual agreement of the readings that come from your hand is more tight as compared to anybody else in your laboratory. Then you are the best analyst in your laboratory. Otherwise, if one day you're brilliant and one day the readings are all scattered, then it shows lack of application, lack of mind. A good professional analyst is not just a good chemist or a biochemist or a microbiologist, but he also knows his instrumentation inside out. He knows the strength of the instrumentation, the weakness of the instrumentation. He knows what all can go wrong. And if a reading comes which he is not very satisfied with, then he knows how to do further analysis to identify what exactly is happening. We will be seeing an example later where for the normal eye, untrained eye, a peak looks normal, pure. But the analyst decides to do spectral evaluation and shows that one peak is impure and the other peak is pure. This is what is expected, the level of professionalism. Fine. Now I'm going to place before you poor practices that have been observed and noted. And one article is about bad laboratory practices by P.T. Kissinger. It can be downloaded from the net. And the other source for me is Dr. Geeta Iyer's book on good laboratory practices, where she talks about common errors in the laboratory and the correct way to do things. But having said that, this is a very small list. You have to draw on your own experience to add to this list. I'm talking about a very limited number of instruments. There are ever so many other instruments. PCR, qualitative, quantitative, real-time, ELISA, FTIR. You have a list of your equipment. It would be a good idea to add to this list and keep it near your place of work. Use signages to say, caution, avoid these errors. It works wonders. Don't you realize when you're driving on a highway and you say, caution, accident-prone zone, you automatically slow down. That is the power of signage. Let there be a constant reminder in your laboratory that there is no room for mistakes. So, I start with Dr. Kissinger's views on what he has observed in the laboratory, what he calls bad laboratory practices, but I would rather call them poor measurement practices. Including data falling outside the validated concentration range of the assay. Not diluting subsamples or extending the range, but simply reporting data as observed. When the reference standard itself is only 98% pure, reporting the accuracy of the final results to 5 significant figures to placate the quality assurance. What is really important is the number of significant figures in the data, not how many decimal places the software prints out. Assuming the numbers on a mechanical pipette are accurate to better than 0.1% without bothering to calibrate or verify. Using a pH meter to measure or adjust the pH of LC mobile phases that are mixtures of water and acetonitrile or methanol, because making the measurement only with the aqueous components of the mixture is inconvenient. The date on which the calibrating buffers were purchased are not marked on bottles. The temperature of an LC column not controlled for the best precision and accuracy, thinking that temperature control is important only in GC, not in LC. When following a printed method, not paying attention to the salt form of the reference standard, weighing out the same amount, whether the salt is a hydrobromide or a bitartrate. When taking a reference standard out of a deep freeze, example minus 80 degrees centigrade, opening the bottle immediately without realizing that moisture will condense on the standard. If software can give a chromatographic peak area or peak height to 13 significant figures, believing that variation in the 10th or 11th figure will impact on whether an approved drug is really going to be safe and effective, without bothering to understand whether the data itself is intrinsically good or not. Believing an electronic signature based on an 85-digit password is sure to overcome all possibility of fraud. Picking an LC mobile phase pH close to the pK of the analyte without bothering about the effect on shift in retention times if the pH varies slightly from one mobile phase batch to the next. Injecting analytes into the liquid chromatograph using a solution that has a much stronger eluent strength than the mobile phase itself. For gradient elution, forming the gradient using 100% aqueous and 100% organic in each mobile phase bottle, not realizing that outgassing will produce a noisy baseline. When using a fixed loop LC injection valve, loading the valve with a volume that matches the volume of the loop exactly, believing that no sample should be wasted. To determine the lower limit of quantitation for a derivatized analyte in a bioanalytical sample, preparing the derivative in milligram quantities and then diluting it by several orders of magnitude. When studying protein binding for highly bound drugs using dialysis, determining the drug on the protein side because the chromatographic peaks are larger. Believing that LC-MS-MS is the ultra-selective analytical cure-all and you can forget the need to do sample cleanup or chromatography, ignoring matrix effects, ion suppression and labile metabolites that give the same M to Z transitions as the parent compound. If a method works well in one species or matrix, running it with any kind of sample expecting they will all behave the same way. Preferring scientifically elegant methods over simple, practical approaches. Believing it is most important to impress your colleagues with the complexity of your methodology rather than actually coming up with something that works. An assay with 25 steps is to be preferred over one that achieves the same end in 5 or even fewer steps. Now, here is a list of poor practices by Dr. Geeta Iyer. No relevant authorized copies of SOPs, using SOPs which are not revised and updated, using instruments which are not validated, not using calibrated glassware, adjusting the pH of the LC mobile phase, that is a mixture of H2O and a solvent, not making the date of purchase and date of opening of reagent and chemical bottles, not controlling the LC column temperature, backdating records, when taking standards out of refrigeration, weighing it immediately without letting it come to room temperature, not degassing LC mobile phase, in LC injection valve, loading the valve with a volume same or less than the volume of the loop, not recording data directly on the appropriate form of notebooks, writing on scrap, paper napkins, etc., overwriting, and using outdated reagents and solutions. So let us remember, one important challenge for an analytical laboratory is staying in regulatory compliance while maintaining maximum output. Simultaneously, productivity and compliance can only be achieved by laboratories that have competent chemists, high quality managers, and a strong QA team. Analytical laboratories are not immune from unexpected problems like power failures, instrument breakdown, accidental spills, etc. These, however, don't cause regulatory problems since the reasons for repetition of the measurement can be justified. It is the unexplainable data like out of specification results that result in loss of productivity and regulatory problems. A QA system that includes regularly scheduled equipment calibration coupled with monitoring tools will be able to determine whether bad data are due to analytical system problems, whether it is equipment or methods, or simply chemist error. Analytical system problems can be fixed or explained, but chemist errors may or may not be explained in spite of laboratory investigation. Now she talks about wet chemistry, simple thing like weighing, and what are the common errors. Weighing hot or warm samples, weighing materials that lose water rapidly in an open balance pan or in an open vessel. Weighing objects that are too large for the balance pan. Weighing off-center, forgetting to weigh the stopper. Weighing magnetic stir bars and other objects that carry an electrostatic charge. Using a balance that is out of calibration or even out of level. Using contaminated spatula. Spilling samples on the balance pan. An inappropriate weighing range. Weighing of lumps. Not closing balance doors. Returning materials to original containers. So, the suggested correct techniques: make sure that the balance pan and surrounding area within the balance weighing chamber are clean and dry. Use a camel's hair brush to remove any solids. Tear the balance so that the digital readout is as follows: 0.00 plus minus 0.01 gram for a top loading balance, 0.0000 whatever plus minus 0.0001 gram for an analytical balance, and 0 to plus minus 0.00001 gram for a semi-microbalance. Select the proper balance to use as per following criteria. For weighings equal to or greater than 5 grams, use a top loading balance. For weighings between 100 milligrams and 5 grams, use an analytical balance. For weighings less than 100 milligrams down to 10 milligrams, use a semi-microbalance. Dr. Geeta Iyer continues with more suggestions for correct techniques in wet chemistry weighing. Weigh according to the following guidelines. Powders, granules that do not readily lose or gain weight, whether moisture or solvent. Tear weighing paper on the weighing pan. Distribute sample evenly on paper, keeping sample centered on the pan. Record the weight. Solids that readily lose or gain weight, moisture or solvent on standing. Take a covered weighing bottle. Remove the cover, placing it on the balance pan. Add sample to the weighing bottle until the correct amount is indicated on the display. Without delay, place the cover back on the weighing bottle and record the actual sample weight. Liquid samples. Place a quantity of liquid in excess of that needed for the analysis into a container, such as a dropping bottle. Place the dropping bottle containing the sample onto the balance pan and tear the balance display to 0. Transfer the quantity of sample required for analysis into another vessel. Re-weigh the weighing bottle. The sample weight is the absolute weight of the negative number displayed. Common errors in pipetting in wet chemistry: not rinsing the pipette, using pipettes that are dirty or do not drain properly, parallax error, using pipettes with chip tips, blowing out the tip, not wiping the pipette, sucking up liquid so far that it is contaminated by and contaminates the pipette bulb. Suggested correct techniques: make sure that the pipette is clean and dry. Place the tip of the pipette below the surface of the liquid. Using a pipette bulb or other suitable device, suck liquid into the pipette about 2 centimeters above the mark. Allow the pipette to drain completely to a waste container. If any drops cling to the inner walls of the pipette, discard the pipette and use one that drains properly. Suck up liquid a second time to just above the mark and discard the liquid. Once again, suck up the liquid to just above the mark. Wipe the outside of the pipette completely dry. Slowly drop the liquid level in the pipette into a waste container to the point where the meniscus is at level with the mark. Observe at eye level. Wipe off the tip of the pipette once again. Allow the sample to drain into the sample receiving vessel. For non-viscous samples, allow the pipette to drain for about 15 seconds after the liquid has been dispensed. But for viscous samples, allow the pipette to drain for about 45 seconds after the liquid has been dispensed. Do not blow out the pipette or rinse the inner walls down with solvent. Volumetric pipettes are calibrated to deliver by spontaneous drainage. Now, common errors in titrations: dispensing the titrant too quickly from the burette, parallax error, misreading burette volume graduations, acceptance of premature endpoint by not waiting for set time for color to persist, failing to compensate for blanks, bubbles in the dispensing path, especially in case of auto titrators, dirty or greasy burettes. Suggested correct techniques for titration: first, direct titrations. Prepare the sample as directed by the procedure. Prepare a blank consisting of all reagents but not the sample. Rinse a suitable burette with titrant 3 times, making sure that no liquid clings to the inner walls of the burette after the rinses are drained. Fill the burette with titrant to several inches above the 0 milliliter mark. Slowly drop the titrant level until the meniscus is level with the 0 level mark. Observe this at eye level. Make sure that the stop cock fittings are snug and there are no leaks. Make sure that there are no bubbles in the burette tip. Titrate the blank and record the titration value. If the indicator is already at the endpoint color, record the blank as 0.0 ml. Titrate the sample going to the same color as that of the titrated blank and record the volume of the titrant consumed. The sample titration minus the blank titration is the volume of titrant consumed by the sample. Now we talk about back titrations. Follow steps as in direct titrations up to preparation of blank and burette. Add an excess known volume of titrant 1 to the sample and to the blank C. Back titrate the blank with titrant 2 and record the volume consumed. Back titrate the sample with titrant 2 to the same endpoint as for blank and record the volume of titrant consumed. The volume of blank minus volume of sample equals volume of titrant consumed by the sample. Now, electrochemical techniques, common errors in pH, not rinsing the electrode and wiping it dry prior to use, failure to calibrate with buffers prior to use, storing electrodes in distilled or deionized water, accepting a calibration with a slope correction of greater than plus minus 2%, not maintaining a proper level of filling solution, example, potassium chloride in electrodes, storing the electrode dry, incorrect calibration, and use of expired buffers. In potentiometric titrations, errors regarding electrodes and their use, bubbles in burette or burette delivery tubing, titrating too rapidly, errors related to blanks and endpoints. And in Karl-Fischer titrations, using wet methanol, air bubbles in titrant or delivery tubing, not allowing sufficient endpoint delay for complete extraction of water from sample, failure to exclude wet air from the system, using very small or large sample sizes. Now we talk about the more sophisticated instruments and the common errors. Let's take HPLC first. Purging with the column bypass valve closed. Incompatible rinsing, example, not using an intermediary solvent when switching from normal to reverse phase. Rinsing a high salt containing buffer with an organic solvent like methanol without first flushing with water. Not checking for leaks. Over tightening fittings. Flow rate too fast. Not degassing. Ignoring error messages like low lamp energy. Misloading an auto sampler tray. Running out of mobile phase. Failure to perform routine maintenance. Accepting poor chromatography. Here we have common errors in IR spectroscopy. Potassium bromide sample dispersion too dilute or too concentrated. Using a wet sample or wet potassium bromide. Failure to attenuate reference beam. Cloudy salt plates or improper storage of salt plates. In GC, using gas pressures that are too low, not changing septa, over tightening fittings, improper alignment of autosamplers, touching hot surfaces, exceeding maximum column temperatures, accepting poor chromatography, and worst case, running out of gas altogether. In atomic absorption, using a system with low gas pressures, running out of gases, allowing the acetylene primary gas cylinder pressure to fall below 75 psi, using the wrong burner head or wrong flame, using a dirty nebulizer or nebulizer tubing, aspirating standards in the wrong order, using the wrong lamp energy. And in UV visible spectrophotometry, using dirty cuvettes, not using matte cells, wrong blank, using wrong type of cuvettes, using the wrong wavelength, and not zeroing the instrument. Here are some interesting tips for installation and care of HPLC columns. When installing and removing columns, always use two wrenches together, one on the instrument fitting nut and one on the column fitting nut. Insert columns such that the tubing between the columns and instrument parts, such as sample valves and detector inlet, is not placed under undue stress. Insert the column with the flow in the proper direction as indicated by the directional flow arrow on the column label. Never pump mobile phase through a column at a flow rate that would exceed system or column pressure limitations. Never heat a column above its maximum rated temperature. Column fitting should be tight enough to prevent leaks but not so tight that the fittings break. If a leak cannot be controlled after reasonable tightening, cut the tubing and install a new compression fitting. Hand tighten column fittings before applying the wrench. This ensures that fittings are in place and not cross-threaded. Never leave solvents in a column that are at the edges of the rated pH or that contain high concentration of salts. When switching from one HPLC mode to another, such as from reverse phase to normal phase, make sure that all the lines have been flushed with intermediate solvents and the final mobile phase prior to installation of the analytical column. HPLC column, when out of the instrument, should be stored with the end cap snugly tightened. An appropriate storage solvent such as methanol water should be used. An organic component such as methanol is important for preventing microbial growth within the column. HPLC columns, when on the instrument but not in use, should be under a slow trickle flow of about 0.1 ml per minute to keep mobile phase in constant motion. Similarly, for installation and care of GC columns, when installing and removing columns, always use two wrenches together, one on the instrument fitting nut and one on the column fitting nut. When installing fused silica columns, make sure that the ends are squarely cut perpendicular to the column length, not jagged or beveled. This is best done with a ceramic column cutter. Examine the ends using a magnifying glass. Insert the columns the proper distance into injectors and detectors as specified by the instrument manufacturer. Never heat a column without a flow of carrier gas. The flow of carrier gas should not be turned off at temperatures above 400 degrees centigrade. Never heat a column above its maximum rated temperature. Column fitting should be tight enough to prevent leaks but not so tight that the column ferrules break. Hand tighten column fitting before applying the wrench. This ensures that fittings are in place and not cross-threaded. For new columns prior to use, condition as per the column manufacturer's instructions. Make sure that the detector end is not connected during conditioning. GC columns are fairly rugged. When out of the instrument, they can be stored almost anywhere, such as in a drawer or hanging on a wall rack. Now about preparation and standardization of volumetric solutions. Volumetric test solutions should be prepared and standardized as per procedures in standard reference work like the pharmacopoeia. Readily available standard solutions also need in-house standardization. Volumetric solutions need to be given a shelf life at the expiry of which they should be re-standardized. Records of preparation and standardization should include all raw data like lot number of the primary standard, titration values and calculations. The volumetric solutions themselves should be labeled to show name, concentration, date of preparation and standardization and expiry date. Volumetric solution should be stored at recommended storage conditions. Now, just as I mentioned with calibration due date, you give one date, and then if for some reason you're not able to calibrate by that date, and if the instrument is in use the next day, then an auditor could raise an objection. So I had suggested study the stability of the system. If it is say 8 months or 9 months, you say calibration period is due. The calibration is due after 6 months plus minus 4 weeks. The same principle should be applied when it comes down to stability or use by dates of reagent standards, working standards, etc. Carry out a study to establish for yourself the limits to which the standard can really be used. Now, assuming that this can be used for, let us say, 40 days, then you say, fine, use by date or re-standardized by date, 30 days plus minus 5 days. So you give yourself a little bit of time in case you want to use a reagent and that reagent, the fresh stock has not arrived. This is eminently usable and you can't use it because you said use by date is 31st March and this is 2nd of April, so you can't use it. Don't do that. If you know that that particular reagent or standard is usable till the 30th of April, then you say 31st March plus minus 10 days. So you give yourself a little bit of time to see that whatever replacement has to be done or re-standardization has to be done or whatever has to be done is done. You have an interval of time, not a point in time. Here are some safe laboratory practices. Eye protection, chemical splash goggles must be worn at all times in the laboratory. One should wear durable clothing that covers the arms, legs, torso, and feet. Persons inappropriately dressed for lab should be denied access to the lab. To protect clothing from chemical damage, wear a lab apron or lab coat. An open flame may be ignited only when no flammable solvents are in the vicinity. The person lighting the flame must check with others in the vicinity to see if they are working with flammable solvents. Exercise great caution in handling volatile, flammable solvents such as ether, acetone, and methanol. Never evaporate these solvents on a hot plate in an open system. In case of a fire or imminently dangerous situation, notify everyone who may be affected immediately. If chemicals splash into someone's eyes, act quickly and get them into the eye wash station. Eating, drinking, and smoking are prohibited in the laboratory at all times. Never pipette using mouth suction. Avoid inhaling and never taste any chemical in the lab. Never force glass tubing through a rubber stopper. If glass tubing must be inserted into a rubber stopper, be sure to lubricate both the stopper and the tubing. Always remember, hot glassware looks exactly the same as cold glassware. Be careful what you touch. 100 degrees centigrade is equal to 212 degrees Fahrenheit, and it is the boiling point of water. Pay attention to what you are doing. Lab accidents happen most often when you are cleaning up or doing something you think isn't dangerous. Properly label your reactions and containers. A label is not just crossing out hydrochloric acid and writing waste, etc. with a marker. There are specific rules and procedures to follow. Hoods are not meant for chemical storage. If you're not using certain chemicals, put them away. If the hood has to be used for storage of chemicals, find a different hood to run your reaction. Never run experiments in hoods where waste is stored. Do not use or store incompatible materials in the same fume hood. Minimize potential injury. When possible, avoid performing benchtop work immediately across from a fume hood that is being used for waste collection or in some other experiment. Now finally, a list of good laboratory habits. Safety first, last and always. Always follow safety rules. Wear personal protective equipment. Always wear your safety glasses, safety shoes and other personal protective equipment. Wear only appropriate clothing. Each individual engaged in the conduct of or responsible for the supervision of a non-clinical laboratory study shall have education, training and experience to enable that individual to perform the assigned functions. Maintain a summary of training and job description of each individual engaged in the conduct of or responsible for the supervision of a non-clinical laboratory study. Number of personnel should be sufficient for the timely and proper conduct of the study. Report mistakes or suspected mistakes to your supervisor. Read and become familiar with all SOPs and other documents that relate to your work. Take sanitation and health precautions designed to avoid contamination. Wash your hands. Report any illness that adversely affect the quality or integrity of a study, especially contagious ones. Label all the reagents and solutions in the laboratory areas to indicate identity, titer or concentration, storage and expiry date. Store reagents and samples under appropriate storage conditions so as to preserve the purity and stability. Read and observe all labels and signs. Check for quarantine, release and rejected stickers and read all labels carefully. Ensure that material has not expired and that it is the correct material before using it. Warning and signs in your plant should be printed in all languages spoken in the plant. Keep instruments clean, calibrate and standardize before use. Think before you do anything. Don't rush. Always fill in the blanks. All records are for information. If not applicable, write not applicable, your initials and the date. Record results as you get them. Use indelible ink. No pencils, please. Banish correction fluid. Using correction fluid is not allowed. Line through, initial and date all changes. The correct way to make a change is to line through the error once, clearly write the correction above or beside it and initial and date the correction. Never backdate or falsify data. Ensure that the equipment is validated before use. Do not bring food, gum, tobacco, or house plants into laboratory areas. Check your pest control devices frequently. Before signing anything, check for accuracy, completeness, and correct calculations. Print clearly in logbooks and fill them completely. Record data directly on the appropriate form or notebooks. Never write on scrap paper, napkins, etc. Always attach printouts and labels where indicated. Never average out of specification results to obtain a passing result. During inspection, answer all questions honestly and directly. Use validated computer systems, as the real function of the laboratory is dependent on the accuracy and reliability of series of automated devices that control instrumentation, data management, archiving, interpretation, and reporting. If those automated systems fail to properly analyze, receive, store, interpret, summarize, or organize data, the integrity of laboratory can be compromised. If any pest control materials are used, it should be documented. As I said earlier, I have relied on just 2 authors who have pointed out common mistakes made in laboratories. I asked you earlier and I repeat again. You also have made mistakes. You have seen others making mistakes. We all learn from our mistakes. Success teaches us nothing. The sad part is everybody is willing to talk about their achievements and successes, but nobody is willing to talk about their failures. Let not everybody reinvent the wheel. Whatever I may have read out to you now, maybe most of it you already know. Maybe all of it you already know. All I am asking you is add to this list and keep it in your laboratory as part of your training program. Let everybody constantly be aware that the measurement itself has uncertainty. Let's not add to that uncertainty. As I said, there is an error component and there is an uncertainty component. Uncertainty is because there is a doubt, but error is because there is a mistake. Now, it could be a mistake of the instrument, it could be a mistake of the analyst, it could be a mistake of the sample. Any mistake is lacking in professionalism. So, we want the highest level of professionalism in the laboratory. When we give out a number, that number has a certain reliability, repeatability, reproducibility, believability, credibility.